In this lesson, I'd like to focus on the control of gene expression. You have now learned about how DNA replicates and how DNA is transcribed into RNA, and finally, how RNA is translated into protein. What we haven't talked a lot about up to now, though, is the controlling of the process. And one of the large controls occurs really during transcription. So let's take a look at the control of gene expression. Well, it kind of makes sense. If we've got 100,000 genes in our body, not every one of those genes needs to be on at the same time, because it does cost a lot of money energetically, I should say, to create protein. Some genes are transcribed and translated constantly, and these are called housekeeping genes. For example, the genes that are used to keep our cellular respiration machinery running that you learned about in the last unit. But there are many others that are only used occasionally, and we're going to focus on those. Some code for cancer, and these are called oncogenes. And these genes can be activated by a mutagen. So under normal conditions, obviously, the body wants to repress these genes. And to be honest, some of the chemotherapy drugs that we give can help keep these genes from being switched on, so to speak. So let's take a look at gene regulation. Well, genes are like light switches. They can be turned on or they can be turned off. Cells have to respond to environmental signals, and gene regulation depends on the requirements of the organism. So when the organism's environment changes, that can either turn a gene on or switch one off. And that's regulated by transcription factors that we're going to get into here. Proteins that bind to DNA and control whether RNA polymerase transcribes genes are known as transcription factors. And we'll take a look at one particular example in a minute here. They can activate or turn on transcription, as I've already said, or they can repress or turn off transcription. In this example, we're going to take a look at a series of genes that work together in the E. coli, which remember is a bacteria found in your gut. We focus a lot on E. coli because they replicate every 20 minutes. They're really quick replicators, and they're easy to study, and they're really cheap. Interestingly enough, the one group of genes that we're going to look at here in the E. coli is very similar to the group of genes that regulates insulin metabolism and blood sugar regulation in humans. So an operon is a cluster of genes on a chromosome, in this case a bacteria E. coli, that's under the control of one promoter, and we'll look at promoters in a minute, and one operator. So here is the gene that I'm talking about below. An operator is simply a DNA sequence where a repressor, and remember this is a chemical or protein that stops the production of a gene. The repressor itself is a protein that inhibits transcription. So here is our example, the Lac Operon. In E. coli, the Lac Operon is used to make lactose. And lactase, of course, is the enzyme that's used to break down lactose in humans and in bacteria. So from Unit 1, here is a little bit of biochemistry to review it. We have lactose on the left side, and lactase, the enzyme, breaks down that beta, or I should say alpha glycosidic bond in lactose to make galactose and glucose. So this is a pathway that we're going to look at in the bacteria. Bacteria's favored food source is glucose. If glucose is present, then the operon is off. Again, here is a picture of the Lac Operon, and I'll just quickly identify the important parts. Firstly, there's a promoter region, and this is the recognition sequence for all the genes that lie downstream from it when transcription occurs. The yellow part is actually known as the lacI repressor gene, and it is responsible for generating the repressor protein that will eventually turn off this group of genes if required. Here is a promoter for the structural gene, and in the case of bacteria, there are 3 structural genes that have to be turned on or off when we deal with the metabolism of lactose. And I'm not too worried about what they're called. One of these structural genes codes for Beta-galactosidase. The other gene codes for permease, and the third codes for transacetylase. Again, not important, but these 3 genes right here need to be turned on if lactose is going to be broken down. That's the important part to understand here. And again, this kind of highlights what I just said. There are 3 genes in the Lac Operon that code for the metabolism of lactase, beta-galactosidase, permease, and transacetylase. The lacI gene, which is a sequence upstream, codes for the repressor protein, which we're going to call the lacI protein. So let's look at the situation where lactose is not present in big amounts. And what that means is the bacteria doesn't want to break that down because it's already in short supply. So it's protecting what it's got inside. In this case, the system needs to be turned off. The lacI made repressor protein binds to the operator. RNA polymerase cannot make messenger RNA, and therefore the lactase does not get produced to break down lactose. When lactose is present, then the system is turned on. And that's simply because the bacteria has more than enough of the sugar inside it, and it's good to metabolize the excesses. lacI protein in this case is inactive. Therefore, polymerase can actually make its mRNA, and we can break it down. Let's take a look at the absent scenario. So under this situation, the bacteria is low in sugar, and we can see what happens. Well, number 1, the repressor protein that gets produced from the lacI gene prevents transcription to occur because it actually binds to the operator region, which is just upstream of these 3 genes that code for the enzymes, and it blocks, ironically, RNA polymerase from reading along that DNA and transcribing those 3 very important genes. So this repressor protein right here kind of just blocks the way like a roadblock and stops RNA polymerase from reading the DNA and causing these proteins to be produced. This is the opposite scenario. So in the case where the bacteria has enough sugar on board or lactose present, the opposite occurs. Number 1, lactose itself, which is that disaccharide, binds to the repressor protein that is normally produced by lacI, which is the part of the DNA that codes for the repressor. When the lactose binds to the repressor, it actually stops it from being able to bind to the operator right here. So it's the sugar itself that ultimately turns off the production of enzymes that break it down. So what we can see here is when the operator remains free of repressor, the RNA polymerase can actually continue to transcribe the 3 genes downstream from the operator, therefore producing the 3 enzymes that are needed to break down the lactose. So kind of interestingly, when you think about it, the presence of the substrate, which in this case is lactose, allows this whole process to occur so that enzymes which shut off its production can continue. So it's kind of a neat process when you think about it. Let's recap. The operator, remember, is a binding site for the repressor protein that controls whether or not the Lac Operon is activated or inactivated. The promoter is actually the area on the DNA that allows the RNA to bind to it. And to be honest, this is true for all DNA that codes for RNA. There's always going to be a promoter site, a recognition sequence that allows RNA polymerase to bind upstream from that gene. The repressor, remember, is the protein that normally shuts off a group of genes that codes for a certain product. In this case, the repressor protein shuts off the production of enzymes that control the breakdown of lactose. And finally, lacI is a gene in this case that controls the production of the repressor protein in itself. And lacI is upstream of all the other genes. So there you have it, a good example of transcription control and being able to control the levels of proteins in the body, a smart way for the body to conserve energy when it doesn't need a protein and to use it when it does. Finally, this is just a small look at the 3 enzymes that we've been talking about. Beta-galactosidase, number 1. This is the enzyme that actually breaks down the glycosidic bond between the 2 monosaccharides in lactose. Permease is interesting in a way because it actually makes the cell membrane a little bit more permeable, which allows the lactose to be taken up by the cells instead of being concentrated in the cytoplasm. Ironically, despite us talking about it, transacetylase does not really have a known function. Regardless of that, it is known that all 3 of these enzymes are required to metabolize lactose.